Hi friends, welcome to IGCSE Study Room where you can revise biology topics from the Cambridge IGCSE syllabus. This video summarizes part 2 of topic 6 plant nutrition. In the previous video, we left off at the factors that are important for photosynthesis. Chlorophyll is needed because it helps absorb light. Light is important because it provides the energy to drive the reaction. And carbon dioxide is important because it is converted into sugars such as glucose. Let's look at some investigations that will prove the importance of these factors for photosynthesis. Basically, what we need to do is to get a functioning plant and deprive it of each of these factors individually and prove that the plant stops photosynthesizing when these factors are absent. Starch is stored in chloroplast where photosynthesis occurs. So testing a leaf for starch is a reliable indicator of which parts of the leaf are photosynthesizing. Although glucose is produced in photosynthesis, it is not reliable to test for it because the glucose is quickly used, converted into other substances, and transported or stored as starch. There are two things that has to be removed from the leaves before the investigation. They are starch and chlorophyll. First of all, we are going to destarch all our plants by leaving the plants in the dark for 48 hours. During this period, the plants will be unable to photosynthesize and therefore use up all its starch for respiration. This means that in the beginning of our experiment, all plant test and control will have absolutely no starch in them. So after the experiment, if we do a starch test with iodine and we find starch is present, it would indicate that photosynthesis has occurred. So as you can see, leaving the plants in the dark place will help remove all the starch from the plants. Next, we need to remove the chlorophyll to decolorize the leaf so that the color change from iodine is easier to see. As shown in the illustration, the color change is easier to determine when the chlorophyll is removed from the leaf. Also, we can't just add iodine onto a fresh leaf and expect results. First of all, we need to break the leaf so that the iodine can seep in to begin with. These are the steps to remove chlorophyll. Boil the leaf in water and make it permeable. Boil the leaf in ethanol to remove the chlorophyll from the leaf and to decolor it. Rinse the leaf in water to soften it. The leaf is now ready to be tested for the starch with iodine solution. The first investigation we'll look at is the need of chlorophyll in photosynthesis. To prove that chlorophyll is needed for photosynthesis, a variegated leaf may be used. Since it is a type of leaf that is partially green and partially white, that is, only parts of the leaf has chlorophyll. First, the leaf is put in boiling water to kill the cells and break down the cell membranes. This makes it permeable. Next, the leaf is put for 5 to 10 minutes in hot ethanol in a boiling test tube. This removes the chlorophyll so color changes from iodine can be seen more clearly. Then the leaf is dipped in boiling water to soften it. After that, the leaf is spread out on a white tile. An iodine solution is added to it. The white areas of the leaf contains no chlorophyll, and when the leaf is tested, only the areas that contained chlorophyll will stain blue-black. The other areas that had no chlorophyll will not turn blue-black. Since no photosynthesis occurs here, and so no starch is stored. Next, let's investigate the plant's need for light in order to photosynthesize. Just as with the previous experiment, the plant must be first destarched by leaving it in a dark place for 48 hours. Then partially cover a leaf of the plant with aluminium foil and place the plant under sunlight for a day. The leaf can then be removed and tested for the starch. However, we must first do steps 3 to 5 like in the previous experiment and prepare the leaf before the iodine test. These steps are carried out to make the leaf permeable, to remove the chlorophyll from the leaf and to soften it. The leaf is placed on a white tile and tested for starch using iodine solution. The covered areas will be deprived of light, whereas the rest will be exposed. The area of the leaf that was exposed to sunlight will turn blue-black, while the area that was covered with aluminium foil will not turn blue-black, as it did not receive any sunlight and could not photosynthesize. Therefore, starch is not present in that area. This proves that light is necessary for photosynthesis and the production of starch. In the next investigation, we will determine if carbon dioxide is needed for photosynthesis. Destarch two plants by placing them in the dark for a long time. As discussed previously, this ensures that no starch is present in the plant at the beginning of the experiment. Place one plant in a bell jar which contains a beaker of sodium hydroxide which will absorb carbon dioxide from the surrounding air. Place the other plant in a bell jar which contains beaker of water. This is the control experiment which will not absorb carbon dioxide from the surrounding. Place both plants in bright light for several hours. Prepare the leaves as shown in steps 3 to 5 and test both plants for starch using iodine. The leaf from the plant placed near the water will turn blue-black as it has all necessary requirements for photosynthesis. However, the leaf from the plant near the sodium hydroxide will not turn blue-black, it will remain orange-brown, the color of iodine solution, as it could not photosynthesize due to the lack of carbon dioxide. Now that we have established that plants need chlorophyll, light, and carbon dioxide for photosynthesis, let's investigate the factors that affect the rate of photosynthesis. They are light intensity, carbon dioxide concentration, and temperature. To investigate how fast photosynthesis is happening, we may use an aquatic plant that photosynthesizes in water and count the number of oxygen bubbles it releases in a minute. As photosynthesis occurs, oxygen gas is made and released. Since the plant is in water, the oxygen released can be seen as bubbles leaving the plant. The number of bubbles produced over a minute can be counted to record the rate. The more bubbles produced per minute, the faster the rate of photosynthesis. So, for investigating the effect of changing light intensity on the rate of photosynthesis, the setup may look like this. A photosynthesizing aquatic plant is placed in a beaker of water with sodium hydrogen carbonate. An inverted funnel is placed on the plant and an inverted boiling tube is placed on the end of the funnel. A thermometer is used to monitor the temperature of the solution. A lamp is placed near the beaker, and with the help of a ruler, it should be placed at different distances from the beaker. The rate of photosynthesis must be calculated. The resulting data will produce the graph like this. So, initially increasing light intensity increases the rate of photosynthesis until some other factors becomes limiting, after which the rate becomes constant. So, without enough light, a plant cannot photosynthesize very quickly even if there is plenty of water and carbon dioxide. Increasing the light intensity will boost the rate of photosynthesis. However, at one point, the rate of photosynthesis stops increasing. This means that even with a stronger light intensity, there may be some other factors limiting the rate of photosynthesis. This limiting factor may be either not enough carbon dioxide to make the rate of photosynthesis faster, or temperature not being high enough. Next, let's investigate the effect of changing carbon dioxide concentration on the rate of photosynthesis. The experiment is set up similar to the previous one, where a photosynthesizing aquatic plant is placed in a beaker filled with sodium hydrogen carbonate solution. An inverted funnel is placed on the plant and an inverted boiling tube is placed on the end of the funnel. A thermometer is used to monitor the temperature of the solution. A lamp is placed near the beaker and the distance of it from the beaker must be kept constant. To change the carbon dioxide concentration, different amounts of sodium hydrogen carbonate may be dissolved in the water in the beaker, and the rate of photosynthesis must be calculated. Remember that care must be taken when investigating a condition to keep all other variables constant in order to ensure a fair test. The resulting data will produce a graph like this. So initially, increasing carbon dioxide concentration increases the rate of photosynthesis until some other factors becomes limiting, after which the rate becomes constant. Therefore, even if there is plenty of light, a plant cannot photosynthesize if there is insufficient carbon dioxide. The factors which could be limiting the rate when the line on the graph is horizontal include temperature not being high enough or not enough light. Now, let's investigate the effect of changing temperature on the rate of photosynthesis. Once again, experiment is set up similar to previous one, where a photosynthesizing aquatic plant is placed in a beaker filled with sodium hydrogen carbonate solution. An inverted funnel is placed on the plant and an inverted boiling tube is placed on the end of the funnel. A thermometer is used to monitor the temperature of the solution. A lamp is placed near the beaker and the distance of it from the beaker must be kept constant. To change the temperature, a hot plate may be used to heat up the solution in the beaker, and the rate of photosynthesis must be observed and calculated at different temperatures. The resulting data will produce a graph like this. The rate of photosynthesis initially increases with temperature, as the number of collisions between enzymes and substrates increases until the optimum temperature is reached, after which the graph reverses and eventually drops down to zero. This is because high temperatures will denature enzymes that are required for photosynthesis. If it is too cold, also the rate of photosynthesis will decrease. So that concludes part 2 of topic 6, plant nutrition. Hope you found this video useful. Thank you for watching. And please don't forget to subscribe IGCSE Study Room for more biology revision videos.